Good morning, everyone. First, let me say thank you very much for inviting me today. I'm looking forward to talk to you about my PhD work, which focuses on an unusual structure that we see in frogs called the urostyle. So, a little bit about myself. I'm currently a postdoctoral scholar at Harvard University. Right now, I study the development of the pelvis, a skeletal structure that is highly unique but incredibly understudied. So it is extremely important to walking, childbirth, and diseases like hip arthritis. So we know very little about how the pelvic girdle forms, the gene that regulates its development, and how its complex articulations arise. So by using single cell and spatial transcriptomic approaches, I'm delving into studying this furthermore these days. But before my postdoctoral work, I joined the University of Chicago for my PhD in 2016, and I graduated last year. I did my undergraduate studies in Sri Lanka at the University of Peradeniya, and I was lucky enough to call my PhD lab, surrounded by so many amazing people, my second home. In Sri Lanka, I worked on comparing skeletal developmental patterns of different lineages of frogs and compared the patterns in ontogeny within an island and the mainland India. So my dissertation work at the University of Chicago focused on the structure, the urostyle. So it is the bone that you see at the end of the vertebral column. Today, I will give a brief introduction into why I studied the urostyle, and then we'll delve into the three main aims I want to discuss today. Over the past few decades, this structure has been neglected as a rod of bone and has received very little attention. Up until today, there are no studies done on why this evolves in frogs or the developmental genetics aspect of it. Basically, my work focused on addressing these gaps in the literature regarding the development of the urostyle. Let's quickly look at what evolutionary novelties are. Novelties, as the word implies, are new things. I just want to emphasize on the two most known ones. One emphasizes the function, essentially focusing on the origin of novel functional capabilities like flight or vision. These are examples of novel functions, whereas the other definitions are structural, emphasizing on the developmental origin of novel body parts. In a novel body part, these would involve changes at the genotypic and phenotypic level. Phenotypic as in morphological changes. And these changes would eventually affect the developmental and adult traits. If we look at the urostyle in the context of the two types of evolutionary novelties that we discussed, the urostyle is called a structural evolutionary novelty. Based on this, my PhD work focused on studying the structure in depth. First part of the work had been to look at the changes that we see at the phenotypic level. Secondly, to see the changes at the genotypic level. And finally, I did a functional assay of the urostyle. That means what happens to the frog body plan when this structure is absent. So I created mutants using CRISPR-Cas9 approaches. I will talk about these sections in depth in the subsequent slides. Let's talk about frogs a little bit. Frogs occupy different ecological niches. Some are seen on the ground near terrestrial habitats, calling from trees. There are also some species that live near water sources, or some live deep underwater, and there are also some that use their hind limb to dig and would live underground. Despite occupying different niches, the frog morphotype is conserved across taxa, both externally, where you see a short, stout body, reduced forelimbs, hind limb muscles well developed with no tail, and a flat, broad skull. And also internally, you see a short vertebral column, posterolaterally oriented pelvic girdle, and the urostyle. So all these characteristics, both externally and internally, as we call it, the frog morphotype, is conserved across more than 8,000 species of frogs. If you recall from the small clip I showed you before, you remember how frogs are seen in different habitats, right? Frog jumping is a unique locomotory adaptation that has enabled successful inhabitation of several diverse habitats. So during jumping, the urostyle is thought to transmit force. However, despite being a conserved structure for more than 300 million years, this has received surprisingly very little attention. So till date, we didn't know why did the Eurostyle evolve or the relationship between the Eurostyle and the Aneuron body plan. What you're seeing here is a froglet that has completed its metamorphosis. I have stained this froglet to depict a bone in red and cartilage in blue. If you focus on the Eurostyle, that's the bone that you see here, right? Even though it looks like one bone at the end when it's viewed dorsally, if you look at the same specimen in lateral view, you can see that it is made of two structures. One is the coccyx, the dorsal component, and the other one is the hypocode, the ventral component. So these two structures fuse together at the end of the frog metamorphosis to form the Eurostyle. I will quickly introduce the two structures independently. The coccyx in frogs, that's the dorsal component, right? It is made of fused caudal vertebrae. However, this fusion of caudal vertebrae has evolved multiple times independently across vertebrates also. In vertebrates, the plesiomorphic state is to have a tail with unfused caudal vertebrae. So this is an example of having a tail with unfused caudal vertebrae. But in some lineages, we see convergently evolved tail loss as well. So with the loss of tail, fusion of vertebrae can be seen. Some of the examples are the ural plate of fish. That's what I have highlighted here in yellow color. And this is made of a fused caudal centra. And another example is the pygostyle or the pygostyle in birds. And of course, us humans and apes also have a coccyx. But again, to reiterate on what I said before, the aneuron Eurostyle or the frog and toad Eurostyle is structurally and developmentally distinct because it has because of the contribution of an ossifying hypocode. So coccyx and the hypocode, they two fuse together to form the Eurostyle. A little bit about the hypocode. So in frogs, this hypocode ossifies, and it is only seen in frogs. So let's consider the ossifying hypocode as the adult state. But an embryonic hypocode, which is a thin rod-like structure, so this is a zebrafish embryo, and this thin structure venter to the notochord. So this is the notochord, and there's a thin rod-like structure that extends to the tip of the tail. That's the hypocode in anamniotes. So this structure degenerates in anamniotes after 10 days, but again, only in frogs, it ossifies at the mid-length at the end of metamorphosis. Current consensus is to believe that amphibian hypocode has an endodermal or superficial mesodermal origin. So I want to highlight here that if it is endoderm derived, it is very surprising that it undergoes ossification later in development. Also have to point out that hypocode is derived from a completely different cell population compared to the rest of the axial column. For a tadpole to change into a froglet, it usually takes about 2 months. Let's consider this the time axis. So the females would lay eggs, and eggs would hatch, and free living tadpoles would develop hind limbs first, and then next the forelimbs will emerge. And eventually they would lose their tails at the end of metamorphosis. So aneuron metamorphosis includes dramatic changes to the tadpole body plan. The Eurostyle, the structure that we are focusing, it forms during this period. So that's the period. So the metamorphic climax is denoted by the period when the tadpole gets its forelimbs until the time it completely loses its tail. So this usually ranges from 6 to 8 days. So the Eurostyle ossification is very rapid and is completed during this metamorphic climax. Another interesting fact is that there is a peak in the thyroid hormone during this time, and it coincides with most of the changes that we see in the frog body plan during metamorphosis. The first part of my work was to study the Eurostyle at the phenotypic level. That means look at the changes that you see morphologically when this structure forms within the tadpole body. So these are some of the methods that I used. So I did basic bone and cartilage staining and looked at the cell differentiation using classical histological methods, and also did a couple of antibody stains I won't show most of them here because they seem too technical, but if you want to know more, I'm happy to talk about it later. And also did micro CT scanning to look at the remodeling of some of the blood vessels. Okay, so bear with me until I go through the morphology part. I know it could be a little boring for most of you, but I think it might help you to understand the molecular section better. So I'll quickly go through some of the morphology slides that I have. So I included this figure here to show you the dramatic changes the tadpole undergo during metamorphosis. So this one here is a tadpole right before metamorphosis, and you can see there is a lot of cranial cartilages, right? Cartilage again is shown in blue, and most of the axial column is formed except for the urostyle. So this is where the future urostyle would form. And once the tail is still present. So again, this is before metamorphosis. So once the metamorphosis is completed, the larval cartilages are completely lost and remodeled, bones formed, tail is lost, and the urostyle is completely formed. So yeah, so one of the structures that form at this time is the urostyle, and all these changes happen within 6 to 8 days. I will quickly show how bone and cartilage development during the urostyle formation more closely. So top row are lateral views of tadpoles that undergo metamorphosis. The arrow indicates the time axis. If stage 58 is day 0 of metamorphic climax, stage 66 would be day 6. And lateral views are, so I used lateral views because it's easy to study how the hypercode forms. At the beginning of the metamorphosis over here, you can see that there is no ossification where the urostyle would form, right? But as soon as the metamorphosis is initiated, you can see this tiny ossification venture to the notochord. This is what we call the hypochord. So it starts to ossify once the metamorphosis is initiated. So as the metamorphosis progresses, it increases in length and later fuses with the coccyx. So that's what I was trying to show here. And also, if you look at the second row, zoomed in view, dorsal views of the selected sections from the top row. I just want to highlight that 2 caudal vertebrae would form and they would fuse at the end to form the coccyx. Even though they are derived from different somites, they eventually fuse together. But yeah, for us, to do the molecular work, it was also important to understand how cell differentiation or what kind of cells are present in these 2 tissue types. This step was the foundation for the molecular work, which I will talk about in a bit. So here I opted for a classical histological approach using eosin and hematoxylin. These are dyes which we use to stain nuclei and the extracellular matrix of cells. Also remember, in all my experiments, I'm comparing before metamorphosis with the after metamorphosis stages to look at how patterns and how development changes across time. So these are all sagittal sections taken from 2 planes, one highlighting the coccygeal tissue and the other one highlighting the hypocaudal tissue. I will show like each individually in the next slide. So I will talk about before metamorphosis first. In all stages, the first column are sections taken from the red color highlighted area of the tadpole. So the top 2 rows are coccygeal tissue, where you see that, so these cells, we call them undifferentiated mesenchyma cells. They would eventually form chondrocytes and osteocytes when they get like signals. So I just wanted to see how cells change across time. Yeah, I won't go into too much detail, but as soon as the metamorphosis is initiated, you can see that these were undifferentiated mesenchyma cells, right? But then they aggregate together and then form chondrocytes and osteoblasts. That's what we see as bones in whole mounds where we stain for bone and cartilage. Yeah, and then they fuse at the end of metamorphosis. Okay. So another way of looking at how structures change is to do antibody stains, where we look at expression of different proteins within the tadpole body. So I showed you in a previous slide how the tadpole body undergoes a dramatic remodeling during the time of metamorphosis, and the pelvic region of the frog is considered the most bizarre. So the next step I wanted to do was to see how does musculature change when the urostyle forms. So to do this, I did a couple of antibody stains. One was MyoD. So that's what you see here. The hormones are stained for the antibody MyoD. So it kind of depicts that at the beginning of the metamorphic climax, the tadpole only has one type of muscle called dorsalis trunci, but as soon as the urostyle forms and at the end of the metamorphosis, like three different muscles form and they all attach to the urostyle. Yeah, I hope this is not too technical, but if you have any questions, I'm happy to take them anytime. Please feel free to interrupt whenever you want to. But these are transverse sections. So I did MyoD staining. So MyoD is the antibody you use to look at muscles, and MyoD and laminin. So these are antibodies that we would usually use to look at how muscle fibers look in sections. But at the beginning of the metamorphosis or before metamorphosis, you see, I'm not sure if we can see it clearly, but the muscle fibers are of a similar width across the body. But when new muscles start to form, like over here, so this one is a newly forming muscle called longissimus dorsi. So it has a smaller muscle fiber width compared to the rest of the muscles. So we were able to identify new muscles by comparing the width of the muscle fibers. Again, all these new muscles were attached to the coccyx. So we were curious about the function of having an ossifying hypochord if it doesn't even serve as a surface for muscle attachment. So just like for muscles, I also looked at how does nerves remodel when the urostyle form. Again, to do this, I also did look at different antibodies that were specific for neurons. One that is commonly used is called acetylated tubulin. So these are hormones where you see axial motor neurons extending to the tip of the tail, but because the tadpole loses its tail at the end of the metamorphosis, the axial motor neurons also degenerate along with the degenerating tail. But when you look at transverse sections, so these are before metamorphosis, at the beginning of the metamorphosis, and end of metamorphosis. So green color is a stain called phalloidin, which stains the extracellular matrix. And DAPI stains nuclei in blue, and tubulin is the stain or the antibody that we use to stain the neurons. If you see here, before metamorphosis, the spinal cord is very visible. You can clearly identify the gray matter and white matter. And then at the beginning of the metamorphosis, coccyx forms, hypogod forms here, and the spinal cord is still there. But at the end, when these two structures fuse together, the spinal cord completely degenerates, and it appears that axons are extending outwards from the urostyle. But these were just done to see how the morphology changes across time. Yeah, this is the last part of the morphology part. So far we talked about how bones and cartilage form, muscles and neuron remodel, but even at the end of it, we still couldn't figure out a function for the ossifying hypochord. Previous studies had highlighted that the embryonic hypochord, remember the long, thin rod that is present in anemiones, plays a role in positioning the dorsal aorta. So we wanted to see maybe the ossifying hypochord also plays a role in remodeling the dorsal aorta in metamorphosing tadpoles. So to look at this, I did a CT scanning of tadpoles across development. Sure. Let me walk you through some of the illustrations so that the whole dorsal aorta, that is a blood vessel, occlusion process is a bit clear. So this is a tadpole before metamorphosis, and I'm highlighting the region where you would see the developing urostyle. So the red arrow points at the dorsal aorta over here, and the hypochord would eventually form dorsal to it, so somewhere around here. So before metamorphosis, there are three blood vessels in the tail of a tadpole. One is the DAV, that's the dorsolateral anatomizing vessel, and another one is called the PCV, posterior cardinal vein, and the third one is the DA, that's the dorsal aorta. So let's see how these blood vessels change across time. So it was hard to look at them by using a stain. So that's why I opted for a CT scanning method where we could visually segment individual structures and look at how they change across time. Okay, so this will be the time axis. And at the beginning of the metamorphic climax, the hypocode starts to ossify between the notochord and the dorsal aorta. So remember, this red thin structure will be the dorsal aorta. And here you can see the hypocode is starting to ossify. So what you're seeing here is a segmented tadpole. So how I got this is I would put a stained tadpole in a micro CT and then get individual TEFs, and then you would segment each TEF to kind of highlight these individual structures. Yeah, so this is a stage 61 tadpole focusing on the area where the urostyle is forming. So this is the hypocode, dorsal aorta, and the other blood vessels. So this is stage 63. This is when the tail regression is initiated. So the area I have highlighted, this part, posterior to the urostyle, completely resorbs at the end of the metamorphic climax. By this stage, if you can see the hypocode has increased in its length and has reached its maximum length. And at the very most posterior end of the ossification, it seems that the weight of the hypocode occludes the dorsal aorta, which eventually leads to blood loss in the regressing tail. So this was not seen or discussed in previous studies at all. So it was very surprising to us that the hypocode had a role in the resorption of the tail in tadpoles. Once again, this is to show that the tail resorption happens very quickly. This is the stage 64 tadpole, which has lost almost half of its tail within less than 24 hours. Again, this is a antibody stain just showing how the muscles are degenerating in the cell, in the tail. So by stage 66, that is the end of the metamorphosis, new veins form, and DA, that's the dorsal aorta, that rearranges, and with two iliac arteries enter each hind limb of the metamorphosed frog. Yeah, I also looked at cell death and cell proliferation around the urostyle when this happens. I won't go into too much detail. So just concluding the morphology part, what did we find out? So we saw how the tadpole metamorphosis involves a remodeling of the larval body and forms new structures, and at the same time loses the tail. So the urostyle forms during this period and is very unique to anurans, and we call it an evolutionary novelty. So we highlight that it is a contribution of two different cell populations, and when this forms, the neurons, muscles around the structure remodels, and we propose that the hypocode appears to remodel the dorsal aorta, and this could be aiding in losing the tadpole tail within 2 to 3 days. So we also say that the urostyle, especially the adult hypocode, plays an important role in the evolution of the anuran body plan. Yeah, I'll move on to the genetic part of my study. So that was to look at the changes at the genotypic level. The objectives of this work was to identify genes and gene regulatory networks of the two tissue types, and to study underlying genes that cause the hypocode to ossify only in frogs. Remember, the ossifying hypocode is not seen in any other vertebrates. Only frogs have this structure. As I mentioned earlier, both tissues undergo endochondral ossification. So despite being derived from two different germ layers or cell populations. Therefore, the third objective was to study the differences and similarities between the two tissue types, the coccyx and the hypocode. So I did a survey of transcriptomics and compared them spatially and temporally. Spatially as in between the two tissue types, and temporally as in looking at the 3 developmental stages that I looked for my morphology study. Okay, to look at the differences in RNA expression across development, I followed this method, which was published in Nature Protocols called GeoSeq. So because the urostyle forms at the mid-length of the tadpole body, it was hard to dissect out the cells of interest. Therefore, by following this protocol, I was able to extract cells that I identified from the morphology part. This flowchart kind of summarizes the steps that I used for this spatial transcriptomic method. In a nutshell, so what I did was I dissected out the targeted tissue block and then embedded in something called OCT. So OCT is a concentrated sugar solution. And then I used a liquid nitrogen to flash freeze it. And then used those blocks to kind of take sections using a cryostat, and then used a laser capture microdissection to dissect out the cells of interest. Because I had a foundation of a good morphology part, I was able to identify the cells that I wanted to target. So I used a laser and then cut off the cells that I wanted to sequence. Yeah, this is just showing how the laser would kind of help me cut the cells of interest. Yeah, again, the top figure is the summary of the experimental setup, highlighting the different types of cells that were targeted. And this slide kind of depicts how the sections that were taken for RNA-seq work look like. So the dissected areas, so for the coccyx, the targeted area was around the spinal cord, right? That's what I have highlighted in yellow color. So I would use the laser to kind of dissect out these cells. And for the hypocord, I took the cells that were ventral to the notochord. Again, using the laser, it was easy to dissect them out than using fresh specimens. This is also a close-up of how the coccygeal tissue would look like when it's zoomed in. So this is what I would dissect out. And again, this is a close-up of the developing hypocord tissue under the 20x eyepiece. So yeah. So once the targeted cells were dissected out, you can extract RNA, and I used a single cell-based PCR and then finally sequence them. Then I also did the bioinformatic analysis where you used pseudo-alignment methods to look at genes that are highly expressed or differentially expressed between the two tissue types. I won't describe these things, but for the RNA-seq analysis, I will show you the results that were obtained from one of the experiments. So what you see here is the principal component analysis, a PCA plot, to identify variance components associated with hypocord versus coccyx across stages. So coccygeal samples are the ones with yellow color margins, right? These ones here. And then hypochordal ones are the ones with purple margins. And here you can see that the coccyx and hypocord cluster together across time and space. So coccyx GS samples cluster here and the hypocord here. Again, the heat map also shows how two tissue types possess two differentially expressed sets of genes. There are major changes in cell composition and development across the three stages in gene expression profiles across hypocord and coccyx. The color code I have used here depict the two types of tissues, coccyx and the hypocord, and also the developmental stages. Again, showing that you can see that hypocord has a lot of blue color here, right? That seems some of the genes are switched off in hypocord compared to the coccyx, just highlighting that these two tissue types are very different when compared with each other. Yeah, I won't go into too much detail again, but this is another way of looking at what kind of genes are important in the two tissues. So this is called a reactome pathway analysis. So what I did was I took the upregulated genes between the two tissues and ran a reactome pathway analysis to see which pathways are highly upregulated in the coccyx and the hypocord. This is a new way of summarizing gene functions of the candidate genes that are specific to tissues. So these central circles that you see here in white color are top-level pathways. And when you insert genes of interest, overrepresented pathways are colored in yellow, like here for coccyx, and purple for hypocord. So through this, we saw that most hypochordal genes, those are the ones highlighted in purple color, are involved in organizing the extracellular matrix, like collagen degradation and formation, activation of matrix metalloproteinases, whereas the majority of the coccygeal genes are involved in neuronal remodeling and modifications. Okay, so the box plot to your left is a summary of up and down regulatory genes in the two tissues. There are also genes that are shared between the two tissue types. Those are the ones in the middle, right? Just showing the numbers, the number of genes, which was expected since hypocode and coccyx both undergo endocrine classification. So this one here, this is an interesting plot. So this is called a volcano plot showing differentially expressed genes in the two tissues during development. So volcano plots are an efficient way of showing comparisons between two components. So we are comparing the coccyx and the hypocode here. So when you look at the coccyx, the genes that were upregulated were very normal. So we see some genes involved in ossification and also some involved in neuron development, etc., along with skeletal muscle genes. Surprisingly, the genes that were upregulated in the hypocode included 3 T-box genes, like a TBX1 here, and then TBXT, and then TBXT2. So it was, which was very unusual. So T-box genes are mostly involved in early patterning of an embryo. So it has not recorded in late stages before. So this was very surprising to all of us. So I'll talk a little bit about T-box genes in a few minutes. So yeah, coccyx and the hypocode. So just showing comparisons and the genes, differences of the genes between the two tissue types. The last part of the genetic study was to see the epigenetic profile. That means to look at, I'm sure you have heard of enhancers, promoters. So what kind of enhancers or promoters are activated during the urocyte formation? That's what I wanted to see as my second last step for the genetic component. So for this, we use a method called ATAC-seq. That's I'll quickly explain what that is. So each unit here is called a nucleosome. So these blue ones you see here is a nucleosome. One nucleosome is made of DNA. So DNA wrapped around histone proteins. So over here, we define tightly packed nucleosomes like these ones as inactive, and then ones that are unwrapped like these ones as active. So this is when a gene is expressed. So when an active region is present, we say that a gene is expressed. So this sequencing method called ATAC-seq has become very popular over the last few years because it uses a low number of cells compared to other methods like ChIP-seq, DNA-seq. So I used a recent ATAC-seq protocol called OmniATAC-seq, which uses a combination of detergents to enhance the quality of data. So you might wonder why I focused on open chromatin regions like these ones. So when a gene is active, the open chromatin regions like these ones denote a regulatory region like an enhancer or a promoter, where a transcription factor would come and bind to this region and activate the gene of interest. So again, I used the same stages and tissues as before, and then compared the RNA-seq results that I showed earlier using the laser capture microdissection with this ATAC-seq data to narrow down some of the candidate genes. So the first step was to dissect out fresh tissues from the targeted stages and to create a homogenized solution. Next, you add like TNF5, which would bind to these open chromatin regions and are fragmented at open chromatin sites. So the cut DNA fragments have sequencing adapters on like these on either sides, and they are usually sequenced at the end. So yeah, then I did some bioinformatic analysis and then did a method called peak calling. So that's how we find what the open chromatin regions are. So in our ATAC-seq experiment, when sequenced fragments were mapped against the genome, so these are the sequence fragments, and they would map against the enhancers or promoters within a gene. So adopting a count-based method, we can define these fragment-rich areas as peaks. So these peak data sets can be visualized using a genome browser like IGV. So usually your data at the end would look like this. And if you see a peak like this, you would try to find if it's a promoter or an enhancer with respect to your gene of interest. So that's what I did for my work as well. So I found some peaks that were closer to the candidate genes that I found from the RNA-seq work. Yeah, so I also compared like tissue specific ones, like hypercode specific ones and coccyx specific ones. And future studies are needed focusing on these functional assays of these regions, maybe like knocking them out to see if it disrupts the formation of the urostyle. So coming to the interesting part, it was unusual to see, remember, high expression patterns of T-box genes in the hypercode. As I mentioned earlier, T-box genes are involved in early mesodermal patterning, but have not been recorded in late-stage tadpoles before. So because we saw high expression levels of TBXT and T2 during hypercoder development, I dug into the literature a little bit to see what is known about late-stage expression patterns in humans. Surprisingly, I saw that the morphological position of the hypercode that we see in frogs resembles a congenital vertebral anomaly seen prenatally in 7 to 8-month-old fetuses or human babies. So this is a 78-month-old fetus, and this arrow here points at the notochord, and you can see that there is an abnormal ossification ventral to the notochord, right? If you look at the frog, remember there's the notochord here, and there is this ossification ventral to the notochord, right? And this region, even in babies, this happens closer to the sacrum, and same in frogs as well. And there were a couple of studies highlighting how mutations in T-box genes or excess expression of T-box genes is the cause for these cells venture to the notochord to ossify in an abnormal manner. So same goes for the frogs as well, right? Remember, high expression levels of T-box genes is the reason that we think that this ossification is initiated in frog tadpoles during metamorphic climax. Another interesting fact is that when this happens, so this abnormal ossification in babies only happen during the last days of gestation in humans, where there is a peak in the thyroid hormone. So even in the hypocaudal ossification in frogs, it is directly controlled by the thyroid hormone, which I will show in a subsequent slide. So, and in some studies, they actually compare the frog metamorphosis with the last days of gestation in humans, because there are so many factors that are similar apart from the thyroid hormone. And this anomaly is also seen, or these babies are born to mothers with diabetes. And interestingly, the frog metamorphic climax, during the frog metamorphic climax, there's a peak in blood sugar levels as well. So this was one of the most interesting findings of my work, I would say. And then I tried to create conditional knockouts, or mutants as you call it, to see how T-box genes are directly affect how T-box genes directly affect the formation of the hypercode, which I will show next. But yeah, this is just showing the expression of T-box genes in the hypercoded tissues. As I said before, so I also looked at thyroid hormonal control in the urostyle formation. So I exposed tadpoles to methimazole, a drug that prevents the secretion of thyroid hormone, to see genes that are directly controlled by this hormone during urostyle formation. So I did a similar set of experiments where I did RNA-seq, ATAC-seq using the same stages, and also compared the morphology. I won't show them here, but I will just end the genetics section by showing a bone and cartilage stained tadpole after the methimazole treatment. Remember, the coccyx is here, but even after 2+ months, the hypercode was not formed in methimazole treated tadpoles because they did not have the thyroid hormone. So it showed that it was directly controlled by the thyroid hormone, the formation of the hypercode. The final part of my work was to see to do a functional assay targeting one of the main candidate genes. To do this, I focused on the T-box genes because they were the most interesting gene that stood out among the rest. I used the CRISPR-Cas9 approach in a nutshell. So CRISPR-Cas9 creates mutations in targeted genes. So if this is the targeted gene, in the presence of guide RNA and a Cas9 protein. I used two experimental approaches to look at the functional aspect of TBXT genes and the hypercode. One was I tried creating frame shift mutants using one cell stage injections. So here I injected a mixture of TBXT T2 guide RNAs into one cell stage embryos and looked at the phenotype of the F1 progeny. I will talk about the first approach and then move on to the second one. This is a comparison of the phenotypes of the wild type and F1 progeny. So these are wild type ones after 3 days, 10 days, and these are the mutants after 3 and 10 days. So wild type ones after 10 days had a tail. You can see this developing tail, but mutants after 10 days did not have a tail at all. So the F1 progeny's survival rate was as low as 5%, and they died after 10 to 12 days. Because of that, we couldn't really raise them till the stage that I wanted to study. So I wanted to get metamorphic climax ones that was after 1.5 months, right? But because they died after 10 to 12 days, I had to come up with another approach to find or figure out how does T-box genes affect hypercode in later stages. So to do this, with the help of the NXR at the Marine Biological Laboratory at Woods Hole, I created a conditional knockout line targeting TBXT in metamorphosing tadpoles. So this was the second experimental approach. I'll try to explain it quickly. So what I did was I injected a plasmid with a heat shock promoter into one cell stage tadpoles and let them grow until metamorphosis. So because there is a heat shock promoter, when the tadpoles were subjected to heat, I created the T-box-G guide RNA to knock out TBXT only at the stages I wanted to study. So the final plasmid was a construction of 4 individual plasmids, so 4 individual plasmids were P1, P2, P3, and P4. So P1 had a U6 promoter and a guide RNA, and then P2 had a heat shock promoter, and then finally P3 had a Cas9 with EGFP. EGFP is a fluorescence marker. And finally, they were all included into a P4 plasmid. So this took some time to get it to work, but the final conclusion was that I was able to knock out TBXT in tadpoles during metamorphosis because I would subject them into like heat, and then that would knock out TBXT at the time point that I want to study. Yeah, so these are just individual plasmids that were used to make the final transgenic plasmid. I'll stop here now because I don't want to go into too much detail about the last part, but I want to conclude this by quickly summarizing what I said. So the fossil record of aneurons contains plesiomorphic forms having a tail with few caudal vertebrae like the Triassic Tridobatrachus, and these were followed by the sudden appearance of fossils with the urostyle and no tail in early Jurassic forms like Prosalurus. However, the lack of intermediate fossils raised the question to when did the hypercode evolve? So through my PhD work, we showed how the hypercode ossification likely preceded the tail loss in aneurons. So I first studied the phenotypic changes of this unique structure, the urostyle, where paraxial mesoderm-derived cells contribute to the coccyx, whereas the endoderm-derived ones or superficial mesoderm-derived ones contribute to an ossifying hypercode. And these two would fuse during metamorphosis to form the urostyle. I found that the ossifying hypercode occludes the dorsal aorta, and then this could trigger the tail reduction during metamorphosis. And the underlying genetic changes revealed that hypercode and coccyx have 2 unique sets of genes. And one of the significant genes that were upregulated in the hypercode were T-box genes. I also show how a peak in thyroid hormone is directly controlled. Thyroid hormone directly controls the ossification of the hypercode, and we hypothesize that thyroid hormone directly acts upstream of these T-box genes in the hypercodal cells, which initiate downstream targets involved in chondrification and ossification in hypercodal tissues. We propose that underlying genetic changes within the ancestral gene regulator network have given rise to a novel structure, the urostyle, sometime during the upper Triassic and lower Jurassic period. Finally, I also hypothesized that the genes underlying the development of the hypercode seem to resemble a vertebral anomaly, and frogs can be used as a potential model to understand this in depth in the future. Yeah, so thank you. There are a few people that I want to say thank you before I end this. So first and foremost, my supervisor, PhD supervisor, Neil Shubin, and also my committee, Jim Hankin, Vicky Prince, Chip, who are at U Chicago and Harvard, and my lab, Shubin Lab, everyone in who was in the lab, and all my friends, and also my family. And finally, there are so many people that I would want to thank, and I'm sure I must have missed a couple. So thank you again for joining the talk. And I'm happy to take any questions.
All right, thank you. Thank you so much, Gayani. Very interesting talk, really wide spectrum study. Very nice to see. I think you fuse like information from paleontology, pathology to next gen genomics and anatomy. It's very interesting. I was talking to these ladies here, like how much work that goes into this one study. Very exciting. I think it's a really nice illustration of going from a very simple system to a very complex, diving deep into a system. I'm talking to the students listening. Gayani, we have 40 in the like visible, but we have about 80 participants in clusters. And unfortunately, we have to knock some people away because we were not sure whether they are actually disruptors or not. But I circulated this to some of the Indian herpetology groups. Unfortunately, some of them we might have accidentally knocked off. Thanks. And now the floor is open for questions. We can take direct questions from the audience as well as from the chat box. Shall I ask? Madam, go ahead. Yes.
No, it's not a question. So,
yeah, Madam.
Yes, thank you so much, Gayani, for that fantastic presentation. It's excellent. And I do not, want to elaborate too much because Sampath, I think, did the honors, and proud to see a Sri Lankan young woman scientist achieving so much. It's not a question, but can you, because you told me that your work at Harvard is a continuation of this project and that it embraces the humans. So the applications of what you did, can you tell us a little bit of that?
It's not
in
a direct
to your,
sorry. Yeah.
In relation to
sorry.
postdoc, yes?
Yeah, I was going to say it's not a direct continuation. I'm looking at the pelvic region of humans, how it develops, because no one had looked at it before, but I'm using similar methods, but a bit advanced this time around because we are looking at single cell, not bulk, like taking a lot of cells and doing it rather than I'm taking one cell at a time and then doing the whole transcriptome thing and then going into depth about human diseases, which I didn't do too much in my PhD. I only looked at that one structure. So this time around, I'm looking at diseases in arthritis and how it affects the pelvic girdle development.
Oh, right. So all the best for your postdoc.
Yeah.
And
thank you,
it's been a pleasure hosting you.
Thank you, Gayani. I'm Deepthi Vikramasinghe, staff member from the Department of Zoology and Environmental Sciences. Thank you very much for this excellent and very informative presentation. And I have a little question. Did you see any differences in developmental trajectories of the aquatic and terrestrial amphibians, the frogs?
So I mostly did Xenopus tropicalis, a lab amphibian, because that's all I had access to. But there are some studies that say that the timing of the hypercode development is different in some of the frogs, but all of them develop at the metamorphic climax. So that's a thing that's consistent across taxa. But my studies were mostly on Xenopus tropicalis. So I didn't really get to compare with other taxa.
Thank you, Gayani.
Yeah.
And good luck to you, Gayani.
Thank you. Thank you, Madam.
Gayani, we have a question at the chat box, but I wanted to
Yeah.
have a question, something slightly different coming from my evolutionary background. You mentioned the Eurostyle and the development of the hypercord and all about 200 to 300 million years old. It's to me kind of in the middle of the development of the vertebrates, right? And so one question is, how come that become a evolutionary novelty? Because I know that it's the middle of the vertebrate development, development of vertebrates, but why you made it a novelty? Because it's to me fairly old. And then moving forward, the loss of tail in apes and hominids, especially the apes, from long-tailed primates to no-tail or very short-tailed apes. Are there any parallels in this? Because if you looked into the embryonic development, hominids still carry a tail in the early fetal stages, and then it lost over time. So, are there any parallels of what you mentioned about the hypercord and the ossification of it, and then the disappearance, kind of disappearance of the dorsal aorta along the road? So is that the same process that now you exposed that is happening in the vertebral fetus and led to like tailless hominids? Or are we talking about two things? I'm asking two questions: the novelty and the
I'll start with the novelty question. So we call it a structural novelty because there is a ossifying hypercord, right? That has not seen in other vertebrates at all. It doesn't matter when it evolved, but the thing to consider is that it is there. So the ossifying of the hypercord is a novel thing. And because it fuses with the coccyx, the structure together is called a structural novelty. Because of that, it is recognized as an evolutionary novelty. Because other vertebrates and other animals don't have it. So I hope I answered the novelty question. And then the human thing. So because we had access to frogs, we were able to see how the dorsal aorta occludes and then how the tail loss regresses pretty quickly, but it is very hard to study that in humans. I think one of the follow-up articles that came up from my work actually said that if someone get a chance to study it, they should actually see if this is the same process that happens in humans as well, because we don't know how the dorsal aorta, if it really affects the tail loss in humans or babies during the tail loss time. But we know for a fact that T-box genes are highly expressed in the tail during the loss of tail in humans, because there was a recent study that kind of highlighted that the regressing tail in humans have high amounts of T-box genes during this time. So everything kind of adds up to what we are saying about human frogs as well. But again, no one has looked at how the dorsal aorta is occluding in the fetus during that development period.
Okay, thanks. There's a question, Gayani, at the question box, chat box. It says, dear madam, how the T-box gene of expression is high in fetus of diabetes mothers. Could you please explain on it? Thank you for wonderful speech.
Yeah, so I didn't really do those studies, but there were papers that highlighted that the anomaly that we see in human babies is mostly in mothers with diabetes. So they had done blood tests to see that the blood sugar level in mothers, and have seen that this ossification is only seen in mothers with high blood sugar levels. So so that's not a conclusion based on my study, but we were just comparing our work with the human work that was already done before. So I hope I answered the question.
I have, I have another one quick question. So are you working on PacBio? Is this PacBio whole genome sequencing, or how you do
this is actually 10x genomics. So they have a new method called multiomics, where you can look at single cell a whole transcriptomic, at the same time single cell epigenomics. And also they have another way where you could take the section and then do spatial transcriptomics, where you look at whole transcriptomic across the tissue using a section, using a frozen section.
This is not dovetail genomics that you are working on, right?
No, it's called 10x genomics.
Okay, and then one quick question. Sorry, I'm not taking anybody's time. I think I'll giving some space for people to ask questions. Thyroid hormone. So the thyroid, how thyroid hormone, the expression of thyroid hormone, because T-box, you mentioned that T-box, the two
So
especially effects.
Yeah, I didn't show those results. So I did the same RNA-seq ATAC-seq for the thyroid hormone prevented tadpoles as well. So we saw that when I did the analysis, I saw that T-box genes were not expressed at all in tadpoles that were treated with methimazole. So that means thyroid hormone is kind of needed as an upstream target for the T-box genes to get activated.
Okay,
But,
so no, my question is that how old expression of thyroid? Are there any understanding of the evolutionary age of these hormones? Because you know, I wonder whether the formation of the structure and activation of the gene, and then the gene and then the activation of it through the thyroid. So are they in the same timeline, or are they coming in different times and forming an state?
You mean the expression of thyroid hormone during frog development?
Not in terms of the evolutionary time scale of T-box, formation of T-box genes for thyroid hormone,
I see.
and the notochord and hypochord, because so these are like three different systems that come together in your context and then performing a trait like a function, right?
Yeah, that I don't know. Yeah, that'll be an interesting thing to look at.
This is Dr. Gayani Senevirathne.
Hi. Hi guys. Yeah, very interesting talk. Thanks so much for that. Just a very simple question since there's time. I just was wondering, so would the toad have a similar mechanism? Yes?
Yeah, they do. I mean, they all have the hypercode. Even direct developers and indirect developers, they all have the hypercode and the Eurostar. So the structure is conserved across frogs and toads. So
is it seen in reptiles? Like, I'm just wondering.
No, not in reptiles. Only in frogs and toads. The Eurostyle is a unique structure. So.
Okay, right. I was just thinking like the gecko when they lose their tail is there something similar?
Yeah, but gecko tails have vertebrae throughout the tail, right? I think they just I think the vertebrae usually breaks, and then that's why it takes some time. In tadpoles, you don't have vertebrae across the tail. So that's why it's very easy. Once you lose the blood loss, it's easy for the tail to resolve within a short time period. But yeah.
Okay, thank you so much.
Yeah, thank you.
Thank you very much for that very interesting and informative presentation. And we are so proud of you, like as Sri Lankans, like we are very proud of you, like how you have come, you have reached sky high in your research field. So proud of you, Dr. Gayani Senevirathne. And I would like to especially thank the audience for your active participation and asking questions. And I'm happy if like the students also can ask the questions in this platform, because this is a really good platform to like interact with these international personalities, and like they can incorporate and they can answer the questions. So, and also I'd like to thank Carter Professor, Senior Chair, Professor Deepthi, Professor Preeti Uragama, and Head of our department, who make this conversation with you and then make that conversation into this successful event. Thank you very much, Madam. And I would like to thank Professor Sampath Senevirathne also for running around and making this work. And finally, thanks again, Dr. Gayani Senevirathne, for this very impressive presentation. And we are really proud of you. Thanks a lot.
Thank you.